Imagine a murder has occurred in a very quiet neighborhood. Police find bloodstains and a very small strand of hair in the victim's shirt. There is no eyewitness, no CCTV footage. Sherlock Holmes, Hercule Poirot, and in fact our ACP Pradyuman is also out of town. Now, in this situation, when the police were thinking, how will they solve the case? Dr. Salukhe enters, solves the case very easily, and he becomes the talk of the town. When the press and media were congratulating him and asking him the reason for his grand success, he smiled and said, भाई साहब मैंने कुछ नहीं किया, जो किया एलेक जेफरी साहब ने किया है, मैंने तो सिर्फ उनका टेक्निक फॉलो किया. The press and the media people, unaware of who Alec Jeffrey is and his contribution in the field of science, looked at each other, confused, and said, who is Alec Jeffrey? What did he do? Let's see who Alec Jeffrey is and the wonderful story behind the discovery of DNA fingerprinting. Alec Jeffrey was a British geneticist. He was not searching for some crime-solving tools. He was rather looking for some inherited genetic markers, that is nothing but tiny sequences present within our DNA that repeat itself in different ways, and it varies from one person to another. He was trying to study these human variations and how he could trace the genes that pass from parent to the offspring. On the morning of 10th September 1984, Jeffrey and his team saw something remarkable. They were analyzing the DNA samples using the X-ray film, and there appeared a pattern of bands. This arrangement was different for different individuals, but it showed shared similarity between the family members. In the words of Alec Jeffrey himself, the entire team knew within 30 seconds that they had discovered something remarkable and extraordinary. The bands which Alec Jeffrey and his team saw that day on 10 September 1984 is what we call as DNA fingerprinting. So let's look into the mechanism and the methods used in DNA fingerprinting. So before going into the methods of DNA fingerprinting, let us see what is DNA fingerprinting. DNA fingerprinting, it is also called as DNA profiling. It is a technique that is used to identify individuals based on their unique patterns, and these unique patterns are found in their DNA. Remember that 99.9% of the human DNA is identical, but that 0.1 variation, it is enough to distinguish one person from another. Now coming to the steps of DNA fingerprinting. First of all, we have to collect the sample. You must have seen in popular crime shows that whenever there is a murder, whenever a crime is committed, the police immediately go and they cordon off the place so that no common public, no common individual can enter in the crime region and they can tamper with the evidence. And after that, the police members, they try to look for the biological material that can contain DNA, and then they slowly put it into the evidence bag. Now the sources, the biological material that contains DNA, what are the possible biological materials that they can get from the crime scene? Either it is blood or hair root or saliva, semen, skin cells. So in forensic cases, the investigators, they look for these biological sources from where they can collect the DNA. After that, the DNA needs to be extracted. We need to isolate the DNA in pure form from the cells in the sample. Now, how can we get this DNA? First of all, if you look into the structure of a cell, there is a membrane. So first of all, we have to rupture the membrane. Since we are dealing with animal cells, so we have to rupture the membrane first. How can we do that? By using detergents. The detergents that we normally use is SDS, sodium dodecyl sulfate. It is a negatively charged detergent. So now we have opened the cell membrane. We have broken the cell membrane. Now we can easily reach to the DNA, towards the DNA. But we have to get the DNA in pure form. That is very important. So any contaminants which might be protein or RNA, we have to remove it. We can remove the protein using proteases, we can remove the RNA using RNases, and then we are going to purify the DNA using alcohol, either ethanol or isopropanol, followed by centrifugation. Then we get a clean sample of DNA that is obtained. The third step is we have to cut the DNA with restriction enzyme. Restriction enzyme, as we know, they are molecular seizers. They will help us to generate fragments of DNA at specific sites. These enzymes, that is the restriction enzymes, they cut the DNA at a specific base sequence, and these specific base sequences, as we know, they are called as the restriction sites. And that will result into variable number of tandem repeats or VNTR. This VNTR, they are the DNA segments that vary in length between individuals, and we are looking for these VNTRs. These VNTRs will create the unique pattern for each and every individual. After it is done, we go for gel electrophoresis. This is step number 4. Separate the DNA fragments by size. That is the purpose of gel electrophoresis. The DNA fragments, they are loaded into the agarose gel, and then we apply electric current. And as we know that DNA is negatively charged. Why DNA is negatively charged? Due to the presence of phosphate group. So DNA will migrate from the negative side to the positive side. Smaller fragments will move faster, creating a pattern of bands. So we get a pattern of DNA fragment which is spread across the gel. Now we have to transfer it to a nylon membrane. This is called as a Sadhan blotting. Now what is the purpose of Sadhan blotting? We simply want to stabilize the DNA fragments on a solid surface. Because for carrying out the further procedure, we cannot rely on that agarose gel. It is very fragile. So we need to transfer it to a nylon membrane or a PVDF membrane, polyvinyl membrane, which is much more stable. So the gel is placed on the nylon or a nitrocellulose membrane for that purpose, and the DNA fragments, it is transferred from the agarose gel to the nitrocellulose or nylon membrane using capillary action. This process is called as the Sadhan blotting, and the purpose is mainly to stabilize the DNA fragment on a solid surface. Then we go for hybridization. Hybridization using DNA probe. We identify, the purpose of this hybridization step is to identify and visualize the specific DNA sequence. So the tool that we use in this case is a radioactive or a fluorescent DNA probe. These probes, they are complementary to the VNTR. That means they will go and specifically bind to the VNTR. They bind only to the specific DNA regions. The last step is autoradiography or visualization. This will help us to visualize the unique DNA pattern. A photographic film or a scanner detects the signals from the probe, and the result is a series of dark bands, like a barcode, isn't it? So we get our final DNA fingerprint. So what began as a mere curiosity in a university lab turned into one of the most powerful tools in forensic science. Alec Jeffrey's discovery is used in criminal justice, paternity testing, and also for wildlife conservation.